My name is Joshua Mugabi. I welcome you to Topic 7, Inheritance and Evolution, Learning Outcome B. Today we are going to assess gene technology techniques, their application in various fields, and the associated ethical implications. The learning activity we are going to do is Part A, which says learners visit any nearby facility specializing in genetic engineering to explore key techniques such as recombinant DNA technology, gene cloning, and polymerase chain reaction. They have to make a report showing their application in genetically modified organisms, GMOs, synthetic insulin production, and vaccine development. So we are starting with recombinant DNA technology, which is genetic engineering. And we are going mainly to emphasize on producing human insulin in bacteria. Item 1. Scientists want to produce a large amount of human insulin for diabetic patients. They isolate the human insulin gene and insert it into E. coli bacteria using a plasmid. Task: explain why plasmids are used as vectors. Describe how the human gene is inserted into the plasmid. Explain how recombinant bacteria produce human insulin. Let's start with explaining why plasmids are used as vectors. Why plasmids are used? They replicate independently. They are easy to modify and can carry foreign genes. Describe how the human gene is inserted into the plasmid. How the gene is inserted. Restriction enzymes cut plasmid and the human DNA at a specific site. DNA ligase joins the human gene into the plasmid, leading to recombinant DNA. Explain how recombinant bacteria produce human insulin. How the bacteria produce insulin? Recombinant plasmid is inserted into bacteria. The bacteria expresses the human gene and make insulin during protein synthesis. Insulin is harvested and purified. Let's now look at creating vitamin-rich rice, gold rice. We are still looking at recombinant DNA technology, genetic engineering. Item 2. Bioengineers want to solve vitamin A deficiency by inserting gene for beta-carotene synthesis into rice grain. Task: state the main goal of this genetic modification. Why are multiple genes required for this process? Explain how rice plant expresses the new trait. State the main goal of this genetic modification. Goal: produce rice with beta-carotene, provitamin A, to reduce blindness and malnutrition. Why are multiple genes required for this process? Multiple genes, beta-carotene synthesis, requires several enzymes for other organisms. Daffodil, bacteria. Explain how rice plant express their new traits. Expression: recombinant DNA integrates into rice genome. Rice plant transcribes and translates the new genes to produce beta-carotene in grains. Now let us go to gene cloning. We are going to emphasize on identifying a gene responsible for disease resistance. Item 3. Researchers discover a plant that resists a major fungal disease. They isolate the resistance gene and want to clone it for further study. Task: explain what gene cloning means. Describe the steps in cloning this gene. State why cloning this gene is useful. Explain what gene cloning means. The meaning: it is the making many identical copies of a specific gene. Describe the steps in cloning this gene. Steps: isolate the target gene. Insert it in a vector, the plasmid. Transform bacteria with the recombinant plasmid. Allow bacteria to multiply and clones of the gene. State why cloning this gene is useful. Uses: study gene structure and function. To transfer the gene into crops for improved resistance. Produce proteins encoded by the gene. Still under gene cloning, we want to look at producing GM livestock, genetically modified livestock. Item 4. As scientists want to insert a growth hormone gene into animals, they need many copies of the gene before insertion. Task: why is gene cloning done before inserting genes into animals? Name a vector commonly used for gene cloning. Predict what happens if a wrong gene is cloned. Why is gene cloning done before inserting genes into animals? Why cloning first? 1. To obtain enough copies for experiments. 2. To increase accuracy and success rate of gene insertion. Name a vector commonly used for gene cloning. The vector is a plasmid. Sometimes viruses are used. Predict what happens if wrong gene is cloned. If a wrong gene is cloned, the desired traits are not expressed. Genetically modified project fails or produces unexpected traits. Now move to polymerase chain reaction, PCR. We are going to emphasize on crime scene investigation. Item 5. A police forensic team finds a tiny drop of blood at a crime scene. They use PCR to amplify the DNA before comparing it to suspects. Task. Explain why PCR is necessary in this scenario. Name the enzyme that allows PCR to work at high temperatures. List the 3 main stages of PCR. Explain why PCR is necessary in this scenario. Why PCR is necessary is that DNA amount is too small to analyze. PCR produces millions of copies from a tiny sample. Name the enzyme that allows PCR to work at high temperatures. The enzyme is Taq polymerase, heat-resistant DNA polymerase. List the 3 main stages of PCR. The stages include denaturation, that is when DNA strand separates, annealing, primers bind, extension, Taq polymerase builds new DNA. Still under polymerase chain reaction, we are going to look at detecting a viral infection. Item 6. A hospital tests patient for viral infection such as COVID-19 using PCR, even when symptoms are mild. Task. Explain why PCR is suitable for detecting virus array. What parts of the virus is usually amplified during PCR testing? State one advantage of PCR in medicine. Explain why PCR is suitable for detecting viruses array. Why PCR is suitable? It detects very small amount of phylogenetic material and identifies infections before symptoms appear. What part of the virus is usually amplified during PCR testing? The part amplified is the viral nucleic acid, DNA or RNA, converted to cDNA. State one advantage of PCR in medicine. The advantages: it is fast, accurate diagnosis. It helps prevent spread. It guides treatment decisions. We are still under polymerase chain reaction. We are studying extinct animals. Item 7. Scientists find ancient bones and use PCR to amplify small fragments of DNA to study extinct species such as mammoth. Task: why is ancient DNA difficult to study without PCR? What does PCR allow scientists to do with limited DNA? Give one challenge with using PCR on old DNA sample. Why is ancient DNA difficult to study without PCR? Why difficult? DNA is degraded and present in tiny amounts. What does PCR allow scientists to do with limited DNA? PCR allows amplification of fragments for sequencing and comparison. Give one challenge with using PCR on all the DNA samples. Challenges include contamination, fragmented DNA, and poor quality templates. We want now to look at the report on the application of genetic engineering techniques in genetically modified organisms, in synthetic insulin production, and in vaccine development. In our report, we shall have an introduction. We shall have the key genetic engineering techniques. We shall have their applications in biotechnology. We shall talk about the impact on society, and then we shall look at the conclusion. Let's now go to the introduction. Introduction. Modern biotechnology relies heavily on molecular tools such as recombinant DNA technology, gene cloning, and polymerase chain reaction, PCR. These techniques have transformed medicine, agriculture, and research. This report outlines each technique and explains how they contribute to the development of genetically modified organisms, synthetic insulin, and modern vaccine. Now we can go and look at the different techniques, how they are applied in the different molecular tools. We look now at the key genetic engineering techniques. The first one is recombinant DNA technology. Recombinant DNA, rDNA technology, involves cutting DNA using restriction enzymes, inserting a desired gene into a vector, such as a plasmid, introducing the recombinant vector into a host organism, allowing expression of the foreign genes. This technique enables organisms to acquire desirable traits that they would not naturally possess. Let's move to the second genetic engineering technique. Gene cloning. Gene cloning is the process of identifying the gene of interest, inserting it into a plasmid, transforming bacteria to create multiple identical copies of the gene, producing large amounts of the gene or its protein product. Gene cloning ensures a stable, renewable supply of specific genes or proteins for research and industrial processes. Let's look at the third genetic engineering technique. Polymerase chain reaction, PCR. Polymerase chain reaction is a technique used to rapidly amplify DNA segments. It involves denaturation. This is the separation of DNA strand, annealing of primers, extension by DNA polymerase. Applications include diagnostics, genetic testing, forensic, research in gene expression, and DNA sequencing. Let's now go to the applications in biotechnology. How are these engineering techniques applied in biotechnology? Application in genetically modified organisms, GMOs. We have the techniques and their application in GMO. The first is recombinant DNA technology. Introduces new genes into plants and animal genomes. For example, insert insect resistance, drought tolerance, among many others. Then technique of gene cloning. This one can be applied in GMOs. It ensures mass multiplication of foreign genes before insertion into the host genome. Then PCR, polymerase chain reaction. Its application in GMO. It confirms successful gene insertion and checks genetic stability. Examples of GMOs created using these techniques. There is BT cotton and BT maize. These were engineered with the Bacillus thuringiensis gene for pest resistance. There is the gold rice, was engineered with genes for beta-carotene production. Herbicide-resistant soybean, altered to withstand glyphosate. The benefits of application of techniques in genetically modified organisms. There is increased yield, reduced pest use, enhanced nutritional content, crop resilience to environmental stress. Now let us look at the application in synthetic insulin production, how the genetic engineering techniques are applied in the synthetic insulin production. Application in synthetic insulin production, the roles of the techniques. We have the techniques and their application in insulin production. We have recombinant DNA technique. The human insulin gene is inserted into E. coli bacteria or yeast. Gene cloning. Bacteria are cloned to produce large quantity of insulin. And PCR, polymerase chain reaction, used to amplify the insulin gene and confirm correct gene insertion. Process overview. Human insulin gene isolated, inserted into a plasmid vector, plasmid inserted into E. coli bacteria, bacteria produces insulin protein, insulin purified and packed for medical use. Outcome. We get cheap, abundant, safe, and effective human insulin for millions of patients worldwide. Now let us look at the application of the genetic engineering techniques in vaccine development. Roles of each technique. We have the techniques and contribution to vaccines. Recombinant DNA technology. This produces antigen proteins. For example, hepatitis B surface antigen. Gene cloning enables large-scale production of vaccine antigens. Polymerase chain reaction detects viral genes, monitors mutations, and aids messenger RNA vaccine design. Examples: hepatitis B vaccine produced using recombinant yeast expressing viral proteins. HPV virus vaccine uses virus-like particles made using recombinant DNA. COVID-19 mRNA vaccine, Pfizer and Moderna. Polymerase chain reaction and sequencing helped identify phylogenetic material and design mRNA constructs. Advantages: there is enhanced safety, no live virus needed, faster production, better control of antigen purity. Now let us look at the impact on society. Impact of genetically engineering techniques on society. Medical. Treats chronic diseases like diabetes. Enables rapid response to pandemics. Improves vaccine safety and accessibility. In agriculture, it leads to reliable food production, reduces chemical pollution from pesticides, increases food nutritional quality. Scientists and industry. It facilitates biomedical research, supports pharmaceutical manufacturing, enables biofuel and enzyme production. Conclusion: recombinant DNA, gene cloning, and PCR are foundation technologies in modern biotechnology. Their application in GMOs, synthetic insulin production, and vaccine development have improved human health, agricultural production, and scientific research. As these technologies continue to evolve, they offer even greater potential for solving global challenges in medicine, food security, and environmental sustainability. Thank you for participating in Inheritance and Evolution. We are on Learning Outcome B, Assess Gene Technology Techniques, their application in various fields and associated ethical implications. We have been looking at learning activity A, whereby we are first looking at the genetic engineering to explore key techniques such as recombinant DNA technology and gene cloning and polymerase chain reaction PCR. And we have also looked at the report about their application in genetically modified organisms, GMOs, and synthetic insulin production and vaccine development. So if this has been very useful to you, please visit me at Mugabi Joshua Biology on YouTube.